Hello everyone, welcome back to my channel Stuti Life Science. Thus in this video, I have taken up a topic which is a cell signaling mechanism present in bacteria or in unicellular prokaryotes. This is quorum sensing. So in this video, I am going to discuss about quorum sensing. So what is quorum sensing? It is a kind of communication system in bacteria. Now bacterial cells cannot talk with each other, right? So they need some form of signaling molecule or some form of interaction with each other. So this process forms this interaction system. Rather, in other words, quorum sensing is the process by which the bacterial cells can interact with one another. Now, how does this interaction takes place? This interaction takes place via chemical signaling. One bacterial cell gives out some chemical signal which is perceived by the other bacterial cells, and in this way they interact with each other. So, there is chemical signaling involved over here. Now, it depends on cell density. The main thing that plays a important role, the most important role over here is cell density. Depending on the cell density, quorum sensing will occur. Rather, when the cell density or the bacterial cell density reaches a critical value, there is this quorum sensing process found. This quorum sensing occurs. Now, depending on this quorum sensing, there is regulation of various processes in bacteria. The main thing is that depending on cell density, quorum sensing occurs, and depending on quorum sensing, we have the turning on of certain genes, or there is gene regulation in bacteria. Now, we are saying when the cell density reaches a critical value, there is quorum sensing, and depending on quorum sensing, the bacteria regulating their gene expression. Rather, there is expression of certain genes which are not expressed in lower cell density, when the cell density is lower. So at higher cell density, we have quorum sensing turning on of certain genes. Now, what do these genes do? What are the functions of those genes? We are getting gene expression. So there must be some function of those genes. Thus, quorum sensing plays important role in regulation of certain processes like virulence, like motility, growth, biofilm formation. These are the processes which are regulated by quorum sensing. Rather, quorum sensing plays an important role in these processes. So, the genes are turned on at higher cell density, which are regulating these processes in turn. Thus, the basic thing is in quorum sensing is that the bacteria can sense the cell density, and they can take certain decisions about turning on certain genes which are playing important role in these processes I mentioned above. So we can say this is a kind of decision making phenomena for bacterial cells. Now we are saying quorum sensing. Quorum means light. So it is a kind of bioluminescence mechanism. Bacteria glows in dark. It gives a glowing phenomena to bacteria. Now, if we look into the regulation, before that, let's talk about the signaling molecule that functions over here. We're talking about chemical signaling. Thus, there must be some chemical signal molecule involved. And these phenomena, especially quorum sensing bioluminescence mechanism, present in Vibrio fischeri. It is a type of bacteria where this process is found. And in this bacteria, we have colony generated by illuminescence, bioluminescence. Now, we have different kind of signal molecules that function over here. First type is AI1 or auto-inducer 1. Second type is AI2 or auto-inducer 2. The third type is AIP2. Now, the signaling molecule that we specifically mention over here is AHL. AHL means acyl homoserine lactone. This AHL is the signaling molecule used by Vibrio fischeri for bioluminescence. Now this AHL is a type of AI1 signaling molecule. AHL used by Vibrio fishery, and it can also be used by, rather it is also used by Agrobacterium tumefaciens for tDNA transfer. Now we come to the mechanism. What happens when the cell density is low? One bacterial cell, suppose, is giving out this AHL. Rather, it has the expression of this AHL synthase gene, which is synthesizing AHL given out by the bacteria, but there are no bacteria in the vicinity. The bacterial cell density is lower. Thus, this signaling molecule cannot be perceived by the other bacterial cells. Rather, there are no such bacterial cells in the vicinity which can perceive this signal. Thus, this signaling mechanism will not work. We will have no illuminescence. Now, when the cell density is higher, what happens? One bacterial cell is suppose giving out this AHL, and this AHL synthesized by one bacteria perceived by the receptors present on the other bacterial cells, and in this way when the AHL gets into the other bacterial cells, they are turning on the expression of more AHL molecules. Thus, this is called a auto-inducing system or auto-signaling mechanism. Why? Because this AHL synthase gene synthesizing AHL, now all the bacterial cells, rather the other bacterial cells have receptors for AHL. They perceive this AHL synthesized by one bacteria through these receptors. Thus, these AHL molecules enter in those bacterial cells and they are turning on the synthesis of more AHL molecules. Thus, we have more inducing molecule and they are turning on this AHL synthesis by itself, by themselves. So, it is called an auto-signaling system. This gives a glowing phenomena to the bacteria. The bacteria glows in dark. Thus, we have the regulation of quorum sensing by cell density. We have seen how this is regulated. Moreover, I have shown the structure of AHL over here. In the bottom left corner, I have shown the structure of AHL. We have homoserine lactone coupled to fatty acid side chain. So, this is the structure of AHL. Thus, in this video, I have discussed very briefly about quorum sensing mechanism, how this is regulated. I will come back with other important topics from the syllabus in the next video. So, this is a very new channel and it is a new venture for me. And if you like my video, do share it with your friends, do share with other aspirants. In this way, my videos can reach more and more aspirants. And don't forget to subscribe to my channel. In this way, I will be able to come back frequently with new videos for you people. So, I am ending this video over here. I will come back soon with other videos. Till then, take care, stay safe, bye-bye.